The central dogma is an attempt to explain how information is transmitted from DNA to all of the different working molecules that we have inside of a cell. So this is what we're seeing here in this image. DNA is our genetic information. So what that means is that DNA contains the instructions really to make anything that we might need to make. And we tend to think a lot of times as the protein being the end result. So those are the molecules that we need to make. That is what will really carry out the functions of the cell. Well, what the central dogma describes is that when we take the information in DNA and we use it to make a protein, we do have this kind of intermediate molecule that we are going to have to transmit that information through. So RNA is an intermediate. At least that's what we think of it in most cases. And so DNA contains all of the information. And then what we're going to have actually occur is we will copy certain sections of the DNA into RNA molecules. And that process right there of copying certain sections of the DNA into RNA molecules, that is what we refer to as transcription. So there's a lot of steps that are involved in that as far as copying, deciding which sections to actually copy, and then the copying process. And then once we get those RNA molecules are going to be what we call translated into protein. And when they're translated into protein, I kind of put translated here in quotes because we call that particular process translation. And so we will have a code, which we call the genetic code, which is going to help us decide what that information in the RNA actually means as far as the proteins that we're going to be producing in the very end. So just to sum it up, the central dogma is this linear explanation of how the information goes from DNA ultimately to the working molecules in the cell, which are the proteins. This is by and large the way we think of it. There are some exceptions to this rule, but this is kind of the general layout of it. So if we look at this a little closer, here we see a DNA sequence. So this is a double-stranded DNA molecule. And as we described in the central dogma, the first step to actually really utilizing the information in the DNA is going to be to actually copy it into RNA. So we're going to copy this into what we call a complementary piece of RNA. And so if we go through and we look at that, when this RNA is made complementary, it's going to be a single-stranded molecule. It's going to run anti-parallel to the template. And so what we're going to be doing is we're going to build something that is complementary to this top strand here. So because that top strand would be anti-parallel to where I put my 5 and my 3' down here. So if we go through and do this, first we would have an A. Next, instead of putting a T, we do want to put a U because this is an RNA sequence. And then I'm just filling in here with the appropriate base pairs, again, looking at the top strand. So again, anywhere you would normally put a T, we want to put a U instead. Okay, so this is the sequence that I have. So this is going to be my RNA sequence. Notice it is single-stranded, and that is what really happens in the overall transcription process. So in the transcription process, we produce this molecule down here, which we tend to refer to as a transcript. And then this molecule is going to get translated into protein. And so to talk a little bit more about how we're going to actually take this molecule and translate it into protein, the way that this will work is that each one of these 3-letter or 3-base combinations, you can think of them as a word. And what we mean by that is that that is something that is going to tell the cell what amino acid and I'm going to abbreviate that AA here, to put into the protein. So we may just have 4 different letters written here, as in A, U, C, and G. But here I have just 4 words, if you will, written, and they all say something different. So they all are a different combination of 3 letters. And so these 3-letter combinations that we see, these are what we call codons. As far as the actual number of them that we have, if you look at it being 3 letters together, if you think about it, we could have any one of the 4 letters in the first position, and by 4 letters I'm talking A, U, C, and G, and then we could get any one of the 4 letters in the second position and any one of the 4 letters in the third position. So if it could be any one of the 4 in each one of those places, when we look at all the possible combinations, we multiply 4 times 4 times 4. That means that in total, there are 64 possibilities, and these 64 3-letter possibilities or 3-letter words are going to be what we call codons. This table that we're seeing in front of us, this is what is commonly referred to as the genetic code. So the genetic code, specifically what it tells us, is it shows us all of those 64 codons, or those 64 3-letter possibilities that we can have, and you see all of those listed here. So if you counted them up, there will be 64 total. And the table here tells us exactly what each one of those means to the cell. Since inside the cell our proteins are made up of 20 different amino acids, and we have 64 codons, there will be some redundancy here. So what that means is just you can have more than one codon, or more than one word that codes for a particular amino acid. And we can see that as we start to look through the table. If you look right here, we've got 2 different codons that both code for this amino acid here, which is phenylalanine. In some cases, we have as many as 6, and we can see that here, that actually code for the same amino acid. So here we have 6 that code for this one, which we call leucine. So there is some redundancy. However, we do say that it's not unclear or it's not ambiguous, because every codon has one and only one specific meaning. So it has one amino acid that it codes for, or we do have 3 codons, and those are shown right here, that do not code for amino acids. So these 3 in particular, they are what we refer to as stop codons, or sometimes they're called nonsense codons. And the job of these is to tell the cell to stop making protein, or in other words, to stop adding amino acids to this particular polypeptide that we happen to be making. So we've got 3 of them that are used for that. Other than that, all 61 of the remaining ones will code for amino acids. We do also have one other unique one, and that is this one down here. And this one is what we call the start codon. So the start codon is always found at the beginning, or in other words, the start of a new polypeptide. We go back, here we have DNA sequence. That double-stranded DNA molecule, portions of it will get copied into an RNA molecule, and that's the transcription process. So here in blue, we can see our RNA molecule that we will be working with, and then what the cell is going to do, and it'll specifically be the ribosome, but what it will do is it will look at each one of these 3-letter combinations, and it will essentially locate it on the table. So if we look up the first one, this means that the first codon tells the cell to put in a methionine amino acid when it's making this particular polypeptide. Then we would look at the very next codon, and this one's GGC, and we would locate that particular one on the table. So for GGC, it's located right here, and we can see that GGC codes for this particular amino acid, which is glycine. And then we would look at the very next one, and UAA happens to be a stop codon. It's right up here. So that would tell the ribosome to quit adding amino acids to this particular sequence. So as I've drawn it, this is very short, obviously, But this would be the polypeptide, which will get folded up to make the protein inside of the cell. So that's a quick look at that linear flow of information, or what we refer to as the central dogma, because we're going to take the information that's stored in the DNA molecule, we will copy it into a piece of RNA, which is going to be single-stranded, and then we're going to translate that using a genetic code table, and that's going to tell us really the language of the cell, or in other words, what each one of those 3 letters combinations, which we call codons, what each one of those is going to ultimately mean. So this would be our polypeptide sequence.